Thank you for the opportunity to share our work today. My name is Naveed. I'm a senior postdoc and a clinical lecturer here in Oxford. And our group has an interest in how disordered innate immune responses contribute to the development of cardiovascular disease. Acute myocardial infarction is an important pathology in its own right, and it's also a model of sterile inflammation. So a vessel supplying the myocardium becomes blocked, and because of underlying atherosclerotic disease, this leads to a strong and a coordinated inflammatory response. So within the first couple of hours, you get millions of neutrophils flooding the peripheral blood. They infiltrate the myocardium, and they can induce further injury to the already ischemic tissues. This is followed by a later mobilization of monocytes, which are mobilized from reserves. They induce pro-inflammatory damage to the injured heart as well, but in the subsequent days, they also induce repair and regeneration. Today, I'll focus on the almost immediate elevation that you see of these peripheral blood neutrophils following a heart attack. In the current paradigm, the injured heart synthesizes very large quantities of chemokines, and it recruits neutrophils from distal reserves such as the bone marrow. But we've identified several issues with this current paradigm. Neutrophil numbers in the blood are elevated almost immediately, so prior to arrival at the hospital, whereas chemokine elevation in the peripheral blood is delayed. It takes up to 24 hours to peak. And this is subsequent to the rise in the neutrophils. Thirdly, if you look at the blood from the coronary sinus, so this is the blood flow within the heart, even following PCI, the elevation in chemokines is really modest, about 0.1-fold. And lastly, prior to the arrival of the neutrophils in the injured myocardium, there's very little evidence of which cells could rapidly synthesize enough chemokines to recruit immune cells from distal reserves, such as the spleen or the bone marrow. Therefore, we hypothesize that the very early mobilization of neutrophils is mediated by mechanisms that are not dependent on chemokines. And today I'll share with you data on how activated endothelial cells rapidly liberate endothelial cell-derived exosomes to mediate mobilization of splenic neutrophils. In Oxford, we benefit from a study called the Oxford Acute Myocardial Infarction Study. So patients presenting with a heart attack are really highly characterized with MRI imaging and repeated blood sampling. In the cohort of patients I've used in this study, similar to other studies, we find that the plasma neutrophil number at the time of presentation is prognostic, and it correlates with the extent of myocardial injury. In these graphs, you can see the number of blood neutrophils from the patients on the y-axis, and then you can see the extent of injury, which is a measurement of edema. And you can also see the measurement of the scar, which is taken 6 months following the heart attack using late gadolinium enhancement. And in both graphs, what we find is that there's a linear relationship. More neutrophils in the blood, the larger the injury to the heart. The rapid elevation in these peripheral blood neutrophils in the immediate hours following the heart attack raises the question of where the neutrophils are mobilized from and how they're mobilized. To investigate this and to determine whether the spleen or the bone marrow contributes to neutrophils to the peripheral blood following a heart attack, I used a mouse model where we ligate the coronary artery, we allow the animal to recover, and then at set time points, we can harvest tissues of interest, such as the peripheral blood, the spleen, the bone marrow, and the heart. And we can determine the neutrophil populations using flow cytometry. We find that there's almost a rapid and immediate increase in the peripheral blood neutrophils, and there's a simultaneous reduction in splenic neutrophil number, but there's no alterations in the bone marrow at this very early time point. Neutrophils mobilize from the spleen really rapidly following myocardial infarction, but how is this coordinated? Chemokines are not elevated at this time point, and that's the current paradigm. So we hypothesize that exosomes, which are these membrane-enclosed particles, mediate the response. So exosomes are a subtype of extracellular vesicle, and there's significant overlap between these different types of extracellular vesicle population. They can be grouped as the small exosomes, microvesicles, and then the larger apoptotic bodies. They carry proteins on their surface, and enclosed within their spheres, they can carry RNA, DNA, and metabolites from their parent cell, and this can alter the cellular status or the behavior of a recipient cell. The data I'll share with you today is extracellular vesicles, which have been isolated from plasma using ultracentrifugation, where we initially pellet cells, platelets, and apoptotic bodies, and then using greater centrifugal force, we can pull down the microvesicles and the smaller exosomes. So we tested the hypothesis that extracellular vesicles mediate the neutrophil response in myocardial infarction. I isolated plasma at the time of presentation, and then 6 months later in the same patient to serve as a control. And what we find is that patients presenting with myocardial infarction have significantly more circulating extracellular vesicles versus a 6-month control. And we can isolate these EVs and you can look at their morphology using TEM, which you can see in this image here, or we can look at their composition. And because our study enables us to obtain repeated blood samples from the same patients, we can look at how the extracellular vesicle response evolves over a number of hours. Using a high throughput protein array, you can look at the EV response in great detail. So here we're looking at EV-associated proteins, such as these tetraspanins, CD9, CD63, and CD81. And there's other EV-associated proteins, such as TSG101, flotilin, and annexin 4. What we find here is that there's a very large quantity of extracellular vesicles, which are within the exosomal characterization or size range prior to arrival at the hospital. So we know that the response is very, very quick. But where are the exosomes coming from? There's many cell types in the heart that can produce extracellular vesicles, and we hypothesize that endothelial cells, which are in very close proximity to the infarcted area, they're bathed in the ischemic environment, and that they may signal immune cell recruitment through liberation of these extracellular vesicles. So to test this, I took the same patients at the same time points, and then we can map the EV response with known endothelial cell markers, such as ICAM1, VCAM1, which you can see on this heat map. What we find is that there's an immediate enrichment for endothelial cell-derived EVs at the time of presentation, whereas other markers such as tissue factor and CD16, which is an immune cell marker, show distinct patterning, which is post-PCI, post-coronary intervention. And the greatest signal comes from VCAM1 at the time of presentation. We also find really highly significant relationships between the number of the EVs, their composition, the extent of injury to the heart in these patients, and the number of neutrophils in the peripheral blood. But in the interest of time, I won't share that data with you today. The accumulating data at this stage was suggesting that endothelial cell-derived exosomes or extracellular vesicles, which are rich in VCAM1, may be important in signaling neutrophil mobilization following a heart attack. So we find that they're enriched in the peripheral blood, but how does this tie together with the splenic mobilization of neutrophils? Do they localize to the spleen at all? And it's very difficult to determine the biodistribution of exosomes and extracellular vesicles in vivo because they're very, very small. But I developed a labeling technique that would allow us to assess tissue distribution over time in the mouse. To determine whether you get endothelial cell-derived exosomal delivery to the spleen, I've used a mouse model of endothelial cell activation in vitro, where we can stimulate with TNF-alpha. We find that endothelial cells produce and release extracellular vesicles under basal conditions. If you expose them to an inflammatory factor, they produce and release significantly more. They show the typical morphology, and they're positive for the EV protein markers. And then similar to our clinical study, we find that there's enrichment for VCAM1 on the surface of the extracellular vesicles. To allow quantitative tracing of these extracellular vesicles from cell culture in the mouse, I took a preparation and then I transfected them with a non-mammalian microRNA MER39. So it's not present in the mouse, and then we can use RT-qPCR to determine the biodistribution and accumulation in tissues over time. So injected an otherwise healthy mouse, took spleen at set time points, and what we find is that there's an immediate delivery to the spleen within a couple of minutes, and that these extracellular vesicles remain detectable within the spleen for up to 24 hours. So we know that the endothelial cell-derived extracellular vesicles, which are rich in VCAM1, localize to the spleen, but do they orchestrate any mobilization? And is the VCAM1 essential in this response? Our group has worked on VCAM1 for many years, principally as a ligand for the generation of imaging particles. But it's well known that immune cells are captured to endothelium through interactions of VCAM1 and the receptor VLA4, which is found on immune cells such as neutrophils. We find that VCAM1 is enriched at the time of injury on the surface of extracellular vesicles, and we hypothesize that the inverse might be also true, where plasma-liberated extracellular vesicles rich in VCAM1 could mediate neutrophil mobilization from the spleen. To test this hypothesis, I employed CRISPR base editing to induce a stop codon in the VCAM1 gene in mouse endothelial cells. So upon application of a TNF-alpha stimulus, they don't express VCAM1, unlike wild-type cells, which you can see in this Western blot here. So I tested the hypothesis that VCAM1 was necessary for neutrophil mobilization in the spleen by taking wild-type mice, and I injected them with wild-type or the VCAM1 knockout endothelial cell-derived EVs at the same concentration. 2 hours later, harvested tissues of interest, and we looked at neutrophil populations using flow cytometry. We find that there is a mobilization of splenic neutrophils to the peripheral blood using wild-type endothelial cell-derived EVs, which are rich in VCAM1, whereas the VCAM1 deficient extracellular vesicles fail to mediate this response, thereby demonstrating a pathway where ischemic tissues can rapidly liberate extracellular vesicles from endothelial cells, decorated in the VCAM1 integrin. They can localize through the spleen, they can alter chemokine regulation, and then mobilize neutrophils. So in summary, acute myocardial infarction induces a peripheral neutrophil response that correlates with the extent of injury. Plasma extracellular vesicles decorated in VCAM1 correlate with the extent of myocardial injury and the robustness of the neutrophil response. We find that they localize through the spleen and that the VCAM1 is essential for mobilization of splenic neutrophils to peripheral blood, and therapeutically targeting this axis may allow us to modulate the neutrophil response and thereby preserve the myocardium in the immediate hours following myocardial infarction. Just to wrap up, none of our work would be possible without support from others. So thank you to Robin, who's our PI, everyone involved in Oxami, our funders, the British Heart Foundation, the Oxford BHF Centre for Research Excellence, and we also receive funds from Wellcome and the Novo Nordisk Foundation. Importantly, I'd like to say thank you to my collaborators who have enabled me to undertake this work. Always keen on working with others who hold the necessary expertise to allow us to test our hypotheses. And thank you for your attention. If you have any questions, I'd be happy to answer them.
Thank you very much, Naveed. That was an excellent talk, something that I know nothing about, but understood. So thank you very much for that excellent presentation. Do we have any questions from the audience? I'm looking at the chat. No, we don't. Maybe I can kick it off then for the first question. So at the start, you were saying about that increased neutrophils cause increased injury. Or is that just a relationship, or is it causal? Is there how strong is the evidence that that is the cause of the injury?
The neutrophils have been largely neglected as a therapy in ischemic injury, especially heart attacks. Their numbers rise very, very rapidly before the patients even get to hospital. So the idea of therapeutically targeting them is challenging. How do you target something that's already had a robust response before your patient presents? What we're learning about neutrophils is actually that they're not just these foot soldiers that are very, very volatile. Actually, they show both functional and transcriptional heterogeneity. And this contributes both to further injury once you have a heart attack, but they also contribute to the healing of the myocardium. So one approach based on what I've told you today is we could get rid of all the neutrophils by using an antibody, but unfortunately that doesn't protect the heart. The neutrophils also communicate with monocytes and macrophages in the subsequent days after a heart attack, and they enable repair by pushing macrophages into an anti-inflammatory phenotype. So just getting rid of them isn't good enough. It's about modulating their response in order to reduce the injury and then push them into a reparative phase, which happens anyway following the heart attack.
Very interesting. Thank you very much, Naveed. We do have a question from the audience. I'm going to read it out to you. Do you think these VCAM1 EVs also play a role in other pathologies? For example, endothelial dysfunction?
So we're in the process of developing platforms where we can now move into larger and larger clinical cohorts. So at the moment, it takes us several hours to prepare one sample. But as we move now into microfluidics-based devices, we'll be able to rapidly isolate VCAM1 populations. And we're hoping that will give us insight into both diagnostic applications and maybe prognostic applications in a range of chronic inflammatory conditions.
Fantastic, thank you.
Thank you.
If I could ask one final question, maybe, what are your next steps? What's the thing you're most excited about doing?
So now that we've identified that VCAM1 is essential in this mobilization response, so now we can bioengineer it. So can we make therapeutic exosomes using the VCAM1 as an anchor to target the immune cells, and then we can encase particular types of silencing RNAs or therapeutic RNAs within the extracellular vesicle to change the behavior of or the phenotype of these innate immune cells. So we can start regulating how they behave very early on following a heart attack. And at the same time, moving towards a diagnostic, prognostic platform. So we know there's a robust response, but how can that tell us more about the individual patient and how they're going to fare in the days and weeks after the heart attack?
Brilliant. Well, thank you very much.